Welcome back, everyone. In this part of the topic, we will be talking about the stomach. So in terms of the learning objectives, they're to be able to list and summarize the features of the stomach and be able to list the three phases of stomach secretion regulation, be able to describe the histology and functions of the stomach and its secretions, and be able to describe the movements in the stomach. And then we'll move on to the small intestine, so be able to describe the movements in the small intestine as well. Then be able to describe the features and histology of the small intestine, be able to list the secretions of the small intestine and discuss their function, be able to describe the anatomy and histology of the large intestine, and be able to list the secretions of the large intestine, and finally, be able to describe movement in the large intestine. We'll start with the stomach and just looking at the different parts of the stomach to begin with. We have the cardia region or the cardiac region, which is the point where the esophagus connects to the stomach. It's called the cardiac region because of its location close to the heart. The fundus then is inferior to the diaphragm. You can see it here. It's sort of dome shaped. And it then gives way to the body of the stomach, which forms the main part of the stomach. The body of this stomach then gives way to the pyloric region, and it's sort of subdivided into the pyloric antrum, which is wider than the pyloric canal. You can see it there sort of narrowing. And you can also see that we have these greater curvatures and lesser curvature there, which are the attachment sites for the omenta, which hold the stomach in place. In terms of openings and sphincters, the gastroesophageal or cardiac opening is present when the lower esophageal sphincter relaxes. So that's that opening to the esophagus, right? Then we also have the pyloric sphincter, which when it relaxes, it will allow chyme to leave the stomach and enter the duodenum. Now, in the absence of food, so when the stomach's kind of empty, it deflates inwards. And the two inner layers, being the mucosa and the submucosa, fall into a large fold called a ruga or rugae. Then when the stomach starts to fill up again, that allows then for the stomach to stretch and those rugae then disappear. In terms then of the layers of the stomach wall, the serosa is the outer layer. It's also known as or visceral peritoneum. Then we have this muscularis layer, okay? You can see here that there is an outer longitudinal layer of smooth muscle. We also have this layer of circular smooth muscle, but because the stomach is a really important sort of chamber for churning food and mixing everything, it also has this extra layer of smooth muscle called the oblique layer. Then we have the submucosal layer, and then finally we have the mucosal layer, and this mucosal layer is in contact with the chyme, right? And we have the presence of these pits called these gastric pits, which are the openings for the gastric glands, and they're lined with simple columnar epithelium. And we're going to talk about these in a little bit more detail on the next slide. Now, these cells of these gastric pits include surface mucus cells that produce a very thick alkaline mucus that protects the stomach lining from acid and digestive enzymes. We're going to learn that protein digestion occurs in the stomach, and these protein splitting enzymes are very powerful. And if you don't have that thick alkaline mucus lining the stomach wall, then these proteases can cause tissue damage. We also have mucous neck cells that produce a different type of mucus. Then over here, you can see these parietal cells here. They produce hydrochloric acid and they produce a factor known as intrinsic factor. And intrinsic factor is essential for vitamin B12 absorption from the small intestine. We also have chief cells, and these chief cells are the ones that produce pepsinogen. Wherever you see ogen at the end of a word, you know that it's a precursor. So this is an inactive form of pepsin. And hydrochloric acid converts pepsinogen to pepsin. And finally, we have these neuroendocrine cells here that are producing and secreting regulatory hormones. So we have histamine that stimulates acid secretion, so hydrochloric acid secretion. We have gastrin-containing cells, which we'll talk about in a while. Gastrin is a locally acting hormone. And we have somatostatin-containing cells that secrete somatostatin, and the somatostatin then inhibits gastrin secretion and insulin secretion. So these hormones are summarized here. We have gastrin, and gastrin is stimulated when there's a presence of peptides and amino acids in the stomach. It has a few different targets, so it acts on the stomach to increase secretion by gastric glands, and it promotes gastric emptying. It also acts on the ileocecal valve further down the digestive tract to cause it to relax, and it also acts on the large intestine to trigger movement in there. We have another hormone called ghrelin, and it is produced in a fasting state. So the levels of ghrelin increase just before we eat a meal. Its target organ is the hypothalamus in the brain, and so it's involved in the regulation of food intake, and it primarily by stimulating hunger and also by stimulating that feeling of being full, so satiety as well. Histamine then is stimulated by the presence of food in the stomach. It acts on the stomach, so it's locally acting. It stimulates parietal cells to release hydrochloric acid and also intrinsic factor. And then the somatostatin, which I mentioned on the previous slide, it is stimulated, or it's produced when there's presence of food in the stomach. Okay, and it acts to restrict all gastric secretions, gastric motility and emptying in the stomach, acts on the pancreas to restrict pancreatic secretions, and it acts on the small intestine there to reduce intestinal absorption by reducing blood flow to the intestines. So this has got to do with the sympathetic axon stimulation. And so in terms then of the secretions of the stomach, remember chyme is the ingested food plus the stomach secretions. Bolus of food goes into the stomach, chyme comes out. Mucus then is produced by those surface and neck mucus cells. Certainly the surface cells produce that viscous or very thick and alkaline mucus for protection from the acidic chyme and also from the enzyme pepsin. And irritation of the stomach mucosa will cause greater mucus production, generally. Now, I'm sure that you've heard of things like gastric ulcers, and the gastric ulcers can be caused by things like non-steroidal anti-inflammatory drug use, or also an infection with Helicobacter pylori. And with the non-steroidal anti-inflammatory drugs, they are mildly acidic, so they irritate the stomach lining, but also they inhibit the formation of this thick mucus lining. So as a result, if you don't have that lining, you get the acidic chyme and the pepsin then being able to come in contact with the mucosal wall and cause damage. And for some people, it's just, mild inflammation called gastritis. But for other people, all of the layers of the wall are actually, eroded. And that allows all that acidic chyme then to enter the peritoneum. And that's when it becomes very dangerous. Intrinsic factor is produced by parietal cells. It binds with vitamin B12 and helps it to be absorbed. And B12 is necessary for DNA synthesis. Hydrochloric acid is produced by parietal cells. It kills bacteria because it's got a very low pH. It stops carbohydrate digestion by inactivating salivary amylase. It denatures proteins and it helps to convert pepsinogen to pepsin. Pepsinogen is packaged in zymogen granules, which are like, this inactive form of the pepsin, and they're released by exocytosis from the chief cells, and pepsin then catalyzes the breaking of covalent bonds in protein. And so this results then in proteins being broken down into smaller peptides, and then further digestion of those peptides will occur further down in the small intestine. Okay, we're going to briefly look at the regulation of stomach secretion. Now, we don't think it's necessary for you to remember all of the details of this, but just to understand that there is a cephalic phase, there is a gastric phase, and there is an intestinal phase. So let's just go back here. With the cephalic phase, so this is, going to involve the brain, right? So let's say you're in a situation where you can smell wonderful food, or you've even got food in your mouth, or tactile sensations of food in the mouth, or just even thoughts of food will stimulate the medulla oblongata. Parasympathetic action potentials then are carried via the vagus nerves to the stomach, where that enteric plexus neurons are activated. This stimulates everything, to get started, right? So it stimulates stomach secretion activity. So hydrochloric acid and pepsin, and also the hormones gastrin and histamine as well. And the second phase then is the gastric phase, and stretching of the stomach, because it's got food in it, or, distension, activates a reflex. This is a parasympathetic reflex. Action potentials are carried via the vagus nerves to the medulla oblongata, which stimulates further secretions of the stomach. And the stretching also stimulates local reflexes that amplify stomach secretions. So while, the cephalic phase is kind of the let's get started phase, this is the real sort of action phase. And then we've got finally the intestinal phase, which is the slowing down phase. So the chyme in the duodenum with a pH less than 2 or containing lipids will inhibit gastric secretions via 3 mechanisms. Firstly, sensory input to the medulla from the duodenum inhibits the motor input from the medulla to the stomach. So you get the, cessation or stopping secretion of pepsin and hydrochloric acid. Local reflexes will inhibit gastric secretions. And then secretin and cholecystokinin, which are another 2 hormones that are produced by the duodenum, decrease gastric secretions in the stomach. Okay, in terms then of movement in the stomach, this diagram shows you it here. It's in an awful lot of detail, so we'll just summarize it here. It's a combination of mixing waves, so 80% mixing waves. Remember, I said that the stomach is like a big churning container, constantly mixing that chyme. And then 20% of the movement are these peristaltic waves. Both the esophageal and the pyloric sphincters are closed. And then you get these really strong waves of peristalsis that, force a small amount of the chyme through this kind of partially opened pyloric sphincter. And so factors that regulate secretion can also regulate stomach motility, and increased motility results in an increased rate of emptying. The last thing we'll mention here is chemical digestion in the stomach, and I have covered this. So we have acidity inactivating salivary amylase, activating lingual lipase. That begins the breaking down of those triglycerides into free fatty acids and mono and diglycerides. Breakdown of protein begins through the actions of hydrochloric acid and also pepsin. There is only one stomach function that's necessary for life, and that's the production of that intrinsic factor. And I spoke about that in terms of it's essential for vitamin B12 absorption. And vitamin B12 is essential for the production of red blood cells and normal neurological function. Cannot occur without intrinsic factor. A total gastrectomy then, stomach removal for stomach cancer then, would require somebody to have vitamin B12 injections because they wouldn't have any intrinsic factor. And the contents of the stomach are completely emptied into the duodenum within 2 to 4 hours after a meal. We move on now to the small intestine, which you can see here, and it's shown in this purple, green and pink, highly coiled series of tubing, right? It's the site of greatest amount of digestion and absorption of nutrients and water. The small intestine pretty much takes up most of the room or the space in the abdominal cavity. And it's called the small intestine because even though its surface area is way bigger than the large intestine, it has a smaller diameter, and that's why it's called a small intestine. It's separated or divided into these three divisions here. So the duodenum being that purple short part, which is 25 centimeters beyond the pyloric sphincter. So duodenum just means 12, so 12 inches or 25 centimeters long. The jejunum then is this green one here, and it's about 2.5 meters long one. It's sort of all uncoiled. And then the ileum is 3.5 meters. And in the ileum, we have these Peyer's patches, which are a specific type of lymphatic nodules. And so these lymphatic nodules then will be involved, in surveillance of any particles that enter this part of the small intestine. So the duodenum itself, this purple bit, curves to the left, and the head of the pancreas, which you're not looking at there, is located in that curve. It contains these major and minor duodenal papillae, which are openings to ducts from the liver and or the pancreas, and we'll get to those in the next recordings, because these are really important accessory organs and glands of the small intestine. Now, because the duodenum is a site of, lots of absorption and digestion, there are these specific modifications to increase surface area in the mucosa and the submucosal layers. We have plici circularis, right, which are also known as circular folds, much easier to pronounce. So if we look here and we zoom in on these circular folds, what we end up is looking at something that looks a little bit like this. Now, these circular folds form a deep ridge in the mucosa and submucosal layers. These epithelial cells lining the mucosal layer then, are modified into these finger-like folds or finger-like extensions of the mucosa that contain that green little lacteal of the lymphatic system, and also blood capillaries as well. Then that's, going to increase the surface area drastically for absorption. But even further then we have what's called microvilli. And the microvilli form this brush border, because when you look at it, it kind of looks like, a brush. And these are these microscopic folds of the cell membranes of these absorptive cells, which you can see here, and that further increases the surface area then for absorption. Now, there are glands of the mucosal layer, and there's also a lot of kind of diverse cells as well. I've spoken about these absorptive cells. They are the cells with the villi and the microvilli. They produce digestive enzymes themselves. They form the brush border and absorb digested food. We'll get to them a bit later on. Goblet cells, they're producing mucus. Then we have all these endocrine cells as well that produce regulatory hormones. We don't expect you to know all the letter of the cells, but things like secretin, things like cholecystokinin, which stimulates the release of pancreatic secretions and bile. And we also have other cells that secrete intestinal gastrin, right? We have granular cells. They're also called Paneth cells, and they may help to protect from, bacteria, because they do secrete this lysozyme. That should say lysozyme, not lysosome. They also have intestinal glands called the crypts of Lieberkuhn, and these are tubular glands in the mucosa at the base of the villi that replenish absorptive and goblet cells. So that's pretty cool. And then we have duodenal cells called Brunner's glands, and these are tubular mucus glands of the submucosa, and they produce this alkaline mucus that they then tip into these intestinal glands that then enters into the duodenum. The jejunum and the ileum then are the more distal parts of the small intestine, and there's a gradual decrease in diameter, thickness of intestinal wall, the number of the circular folds, and the number of villi then the further you get away from the stomach. These two places, though, are also major sites of nutrient absorption. I've mentioned about these Peyer's patches. And the ileocecal junction then is where the ileum meets the large intestine. And the ileocecal sphincter and the ileocecal valve prevent movement of chyme from the large intestine back into the ileum. So this ileocecal sphincter, when it relaxes, that's going to allow the chyme to move on into the large intestine. And then when it's contracted, it's going to prevent movement of chyme from the large intestine back into the ileum. Overall, then, the secretions of the small intestine include things like mucus, protects again for protection. Duodenal glands that produce mucus, also secretin. And then we have these digestive enzymes too that are bound to those membranes of the absorptive cells, so bound to the brush border. Disaccharidases, hard word to say, break down disaccharides to monosaccharides, and then they're ready to be absorbed. Peptidases that hydrolyze peptide bonds to form sort of dipeptides and tripeptides and amino acids, and then nucleases, which break down nucleic acids. Endocrine cells produce regulatory hormones, I've mentioned this, such as secretin and cholecystokinin. And the movement then in the small intestine are those mixing contractions that I explained earlier, so those segmental contractions. We also have peristaltic waves as well that propel the chyme along. The ileocecal sphincter remains slightly contracted until these peristaltic waves reach it, and then it relaxes, allowing the chyme to move on into the first part of the large intestine, being the cecum. Cecal distension, so stretching of the cecum, causes a local reflex and the ileocecal valve constricts. That prevents more chyme from entering. It increases digestion and absorption in the small intestine by slowing down the speed at which the chyme enters the large intestine, and it prevents backflow. And so finally to the large intestine. You're looking at it here. The large intestine extends from the variform appendix, and then it terminates there at the anus. It's subdivided into 4 major regions. Firstly, we have the cecum. And then in a different color green, we have this different sections of the colon. So the ascending colon, the transverse colon, the descending colon, and the sigmoid colon there. And then on through into the rectum and the anal canal. As I mentioned, what seems like ages ago now, that the movements in the large intestine are sluggish. And it takes about 18 to 24 hours for all of that chyme to be converted to feces. But there is some absorption of water and salts in the large intestine. There's secretion of mucus, and there's extensive action of microorganisms. So this is where your gut microbiome is located. And it's such an important feature of the large intestine, because those microbes will ferment food. They'll offer protection against pathogens. They'll also stimulate immune responses, and they'll also make things like vitamin K for us, which is important, say, in things like clotting, right? About 1,500 mils of chyme enters the cecum. 90% of the volume is reabsorbed, and that yields roughly about 80 to 150 mils of feces per day. If we look at the regions of the large intestine, first it was that cecum. It contains the blind sac called the variform appendix, or just the appendix, which is a winding tube, and it's attached to the cecum. The appendix walls contain numerous lymph nodules, which suggests an immune function. And the appendix itself may also act as a bacterial reservoir, because there are bacteria in there to repopulate the enteric bacteria after illness, particularly, if you've had a course of a broad spectrum antibiotic, which wipes out all the normal gut bugs, as well as, the bugs that you're trying to kill. Colon then does have these regions, as I mentioned. The colon also has these unique features, which include the haustra, the tenia coli, and these epiploic appendages. All right. With the tenia coli, these are 3 bands of smooth muscle. The circular muscle layer is complete, but the longitudinal is incomplete. And contractions of these tenia coli form pouches called haustra that gives the colon there its sort of wrinkly appearance. And small fat-filled pouches called epiploic appendages then are, attached to the outside. The mucosa has numerous straight tubular glands called crypts, and the goblet cells predominate, so they're going to be producing mucus, but there are also absorptive and granular cells in this, as in the small intestine. The rectum then is a straight muscular tube with a thick muscular tunic. The anal canal then, the superior epithelium is simple columnar, but the inferior epithelium is stratified squamous. And again, that makes sense in terms of, the protection against abrasion due to, feces. It does contain the internal anal sphincter, which is smooth muscle, and the external anal sphincter then, which is skeletal muscle. And sometimes people do develop a clinical condition called hemorrhoids, and that it occurs when there's vein enlargement or inflammation in the anal canal. And it can be very painful condition, and they can also bleed, and that can cause people a bit of a fright, because they see blood in the feces. Now, the secretions of the large intestine, the main one is mucus. It's providing protection. And parasympathetic stimulation will increase the rate of goblet cell secretion of mucus. We also have these pumps, right, in the large intestine. Now, if you think about your microbiome, the bacteria, will produce acid, and the following pumps remove the acid from the epithelial cells that line the large intestine. So you'll have exchange of bicarbonate ions for the chloride ions of hydrochloric acid, and you'll have exchange of the sodium ions for the hydrogen ions of the hydrochloric acid produced by the bacteria. Bacterial actions do produce gases known as flatus from particular kinds of carbohydrates found in legumes and in artificial sugars like sorbitol. And, the other word then for flatus is obviously farting, okay? And bacteria produce vitamin K for us, which is then absorbed. And the feces then consists of water, undigested food, cellulose, microorganisms, and sloughed off epithelial cells. And finally, movement in the large intestine. So mass movements, not as regular as peristaltic movements, but over much greater areas. They're common after meals. They're integrated by that locally acting enteric plexus. They involve longer segments of the transverse and descending colon. We have local reflexes that are instigated by the presence of food in the stomach and in the duodenum that promote peristalsis and mass movements. The gastrocolic reflex is initiated by the stomach, so food in the stomach, and the duodenocolic one is initiated by food in the duodenum. And this eventually all leads to the process of defecation, and a defecation reflex is involved here. Distension of the rectal wall by feces causes parasympathetic stimulation and relaxation of that internal anal sphincter. This is usually accompanied by voluntary movements to expel feces and voluntary relaxation of the external anal sphincter. And this is kind of facilitated via, abdominal cavity pressure increasing caused by inspiration or breathing in and by contraction of the muscles of the abdominal wall. Okay, everybody, I have finished this part of the topic, and when I come back, I'm going to be talking about the liver, the gallbladder, and the pancreas.